Good evening. It's 11:48, so I'm definitely going to make this video less than 12 minutes because it's episode 4 of the Human Genome Sprapp every single day. I'm going to work on this. It's too important not to. Decoding human life. So I have a detractor, a fan, on one of the message boards that I frequent often who's criticized me a little bit about my Excel work here, and he's saying, hey, I would use a different software application or language or something named Perl. Perl is not a cheap hooker, as I mentioned here below. Perl is an actual language, and Mr. Slow and Go, who I deeply respect for his love of the Allison Rosen podcast, as aka Arian BF, that's a big joke if you know what we're talking about. Now, he's saying that the way I have sequenced is an issue, and how am I going to do the sequencing? I want it not look like a true-false situation. I want to make it look better and filter into the table better, and I will take that good suggestion and I did something with it. And so here's what I did. So looking for genes, right? That's really what the title of this episode should be, looking for genes across all this nucleobase data, looking for a string of genes. I set this up because I didn't have all night to do this, and I was doing a lot of research in other areas. I set this for 50 characters. So you can do a 50 character long gene. Now that is not great because the genes are hundreds of nucleobases long, if not thousands or millions, right? So this version is only going to carry 50, and I know that's not a super duper solution, but it does. It's a proof of concept of how the formula works, and it could be applied to handle thousands, I guess, in just this one file here of Excel. What I did was I wrote a bunch of code here to just grab the sequence one at a time and roll it up into one cell. And you can put like 3,000 or maybe even 8,000 characters actually into one cell. So you could look for a gene that is 8,000 characters long, if you can really get it in there, right? And that's pretty darn good, because one of the things I was looking at chromosome data, and there's 250 million base pairs here on like your average chromosome 1, and we're looking for, I was looking at another chart that I don't show here that showed that of those 250 million base pairs, they've identified like 4,500 genes, approximately 50 characters. So I'm just going to show you how it works with 50. Now I'm going to grab the first 50 characters that are in this genome data set that I have, and I've got 100,000 nucleobases in there, so there's 100,000 rows long, this data, right? So I grab the first 50 nucleotides and I paste them over here, and I paste them as values. So I've got 50 characters, and this is already prepped to handle this. This could handle anything up to 50 characters, so I could also put 40 or 35 or something like that, and you can see the formula is just calculated up. The way it works is this is just a match. They look for these 50 in order, and if they match the 50 that I already had, which are being called right here, these 50 are these 50 in yellow. That's what we just did. If those two cells match, then you're going to get the word match here, and that means we found a match. And then this part is checking for the previous 50 to see, has there been a match? Yes. Okay, if there's a match in here, then call this yes, and that way we now have this whole filtered area that shows us the whole area. Anywhere there's a yes is where the gene is. So I pasted all this down already and already did all this. And then if we go back to the pivot table and we refresh now, this is where it gets really interesting. So what you're looking at is you're looking at the number of instances that this 50 character gene, which is not a gene, I'm sure, but it's just 50 characters we pulled out of this data set. How many times it occurred, and look at how weird this looks. It occurs 24 times in each one of the first 10. Every one of these is 10,000 base pairs. So you got, it occurs 24 times in the first 10,000, 24 times in the second, 24 times in the third, 24, then 23. But it's so repetitive. It's insane. I mean, this is crazy. That 50 character sequence is everywhere in all of this data, and it's almost so uniform. It's like almost all this data is like fractal and self-replicating and self-symmetry. There's so much there. It's all the same stuff. It's crazy. And I already dug into this a little bit, as you can probably tell, because I knew this was going to happen. So I started messing around and figuring out, well, is this really so evenly staggered? And I opened up another cell. I did a little formula here, and this is why I do this in Excel and I don't do it in Perl. I know your visualization could still be done in Excel if your database is in Perl, and our database is going to eventually be in Power Pivot, because Power Pivot is going to be what we can use to do this. Now, so the cool thing is that here, when I flip this around in the pivot, this is the advantage of the pivot table. What's so cool? I grab the sequence number, the actual number in sequence where this happened, and I put it on top. And this is a little formula that says, hey, take a look at when this gene match occurred and then when it occurred the previous time within the sequence. They're all 420 characters apart. I did not plan that. I don't know how. This is insane. They're almost all 420. I think maybe one or two. I bet you could even, they're all 420 characters per. So what does that mean? That means that we're probably looking at a gene that's like 470 characters long, maybe? Yeah, it occurs at, or I guess, no, it's really 420 characters long, I guess. Because it really starts at 1 and the next one starts at 420 because it's 50 less than this. So yeah, really, really interesting stuff. There's so much repetition in here. That's why I feel like we're going to be able to dig in and really gain some information. Insane. So the other thing that I learned today is that chromosomes have locations and they have a naming convention that goes with them, and the French named it because they came up with the word petite and they used the letter P. So your chromosomes look kind of like this stuff up here, right? So you've got your chromosomes here, and they're kind of tied, they're twisted, ravaged together, and so even though I'm looking at chromosome 1 right here, it's really tied together in general. And so there's a where it meets, it's called like microtubules, I think. Wherever they meet, there ends up, it's not right down the middle, so there ends up being a long end and a short end to the chromosome, and the short end is signified with a P in the naming convention, and the long end is signified with a Q. So you end up with a naming convention that looks something like this. Like, say we're on chromosome 12. I have no idea where we are in this data. I'd like to figure that out. I got to talk to Mr. Excel, see where he grabbed this from, because I don't think he got it from me. So 12, say we're on 12 and we're on the long end of the chromosome, it would be a Q, and then there's another number that is related to the number of bands. There's the way they do a lot of DNA testing or chromosome mapping is they put dye on the chromosome and certain areas become darker and band, and they number those bands or stagger those numbering of the bands, and that's what the second number designation in the chromosome location is. So say chromosome 12 on the Q part of the chromosome, the longer part, we're at band 23 point, and then you can go off into decimals where they are even finer detail of what they're looking for. So we need to add a chromosome area. Now this would all obviously be on the same chromosome because this is a sequence and ordered, so this is going to probably all be the same part of the chromosome. Eventually maybe you get out of the Q23 10 part, it'd be Q23.11, Q23.12, or maybe Q24. I don't know. We'll have to figure it out. Either way, we're going to have to start naming this in our table so that we can bring in our chromosome locations and have it comparable with other data that's already out there. So those are two things I learned today, and it's before midnight. I got to get this published because I said I'm doing this every day. But that is my answer for finding genes, up to 50 characters for now, up to hundreds, if not thousands. I could really get in here in one cell, but we're going to have to do better than that in Power Pivot to make this sucker work. So we are going to figure that out. And for all the people who love Slow and Go and love me, if you like this genome thing, then go rate Arian BF 5 stars. Go help Allison Rosen out, that poor girl. She's running a terrible podcast and needs all the help she can get. So there are dual goals here with the Human Genome Sprapp today on 2/24/13. So there you go. Can't stock out.